In a word, in nutshell, we can see the neuromuscular transmission impairment disease or disorders. So first we will talk about our autoimmune disorders. Here basically we will cover two types of the disease. First is the myasthenia gravis. And second one, that is Lambertitan myasthenic syndrome. Myasthenic syndrome. So then after discussing, we'll just compare and contrast in between these two diseases. Okay. So first, if we talk about the myasthenia gravis, then in these cases I already said you, just have a very fast recap. I already said you whenever there will be like what? This is the motor nerve ending plate, okay? And here is the, just like those muscles, muscular crest. There will be like the receptors for the acetylcholines. Just like this. So in this case, we have seen what that in this the vesicle, so acetylcholine will be formed. And there will be one type of the protein here, that is known as the semiactoglobin, or already said, and that is the acetylcholine that is stored within the vesicle. Okay. Now this is brevi, I am writing briefly. Then this will be attached, with the another types of the proteins, that is syntaxins, where the calcium will get bind. This is the calcium channels through which the calcium will come in. You will not understand if you don't see the first video. Okay. And before that, the sodium, of course, that will also come in. Now the thing is here, we need to know the what type of the problem do we see in these cases. Okay. And of course, you have to know one more thing. Now, in these cases, we see that 85% of the peoples, those who are suffering from the myasthenia gravis, they will have different things, and the rest 15% will have the different things. So first 85% of the peoples, they will have the problems, or they will have the formations of the autoantibodies. Okay, let me show with another color. I am showing this part here, okay. Just assume this is the muscular parts. Now we already seen what there will be the receptors for the acetylcholine here. Okay. Now what type of the problem we will see in these cases? The formations of the autoantibodies. Okay, in 80% of the people, 85% of the people, they will have the autoantibodies against this acetylcholine receptors. Is the acetylcholine receptors. Now, in these cases, we will see there will be the formation of autoantibodies against them. Okay, autoantibodies. We will talk about in these things in details with a bit. And the rest 15% of the people, they will have the problem with another things like before that, just let's have a concept regarding this thing. Otherwise, you will not have the great idea. Now, another types of the protein that is present on the crest or in the neurolemma, sarcolemma, that we will see. Sarcolemma, that is known as another types of the protein that is known as mask protein. Now, this mask protein, that is the full form of it is muscle specific. I am saying briefly, muscle specific tyrosine kinase receptor. This is the muscle specific tyrosine kinase receptors that is present here. Now the function for this is that will regulate the number of the acetylcholine receptors here. That means, you can say Whenever the acetylcholine is coming here, let's suppose the antibody is not here. For example, for a while, just trust me what I am saying. This acetylcholine is coming, okay. After that, it is coming here, it is getting attached. Then after the functions, we will see there will be like destructions of this whole things, okay. Then instantly, just to replace these things, just to make the new receptors, the function for this mask protein, that will, whenever this protein will be like just like this, whenever it will be destroyed, then we will see this protein or the mask protein that will helps to form again the new receptors here. So the main functions for this mask protein is here, that it will regulate the formations of the acetylcholine receptors on the crest, muscular crest here, okay, muscular crest part. Now we'll see in the 15% of the people, they will have the antibodies, or 20 antibodies against this mask protein. They will have the antibodies against these mask proteins in these cases, okay. So these are the different types of the causes in the neuromuscular transmission, basically especially whenever the autoimmune disorder is concerned. So I am repeating again, in myasthenia gravis, 85% of the people, they will have the autoantibodies against these acetylcholine receptors, this one, okay. Then if this is blocked, then the no, if it is blocked, then the acetylcholine cannot get bind with these things. As a result, there will be no nerve impulse, there will be no, there will be no signals that will be sent to the muscular activities. As a result, there will see the impairments of the muscular function. And the second part of the 15% of the people will see there is another protein. I said this mask protein that is also present on the surface of this muscular part, and this mask protein is responsible for its main function is to regulate the productions and the destructions of this acetylcholine receptors. Whenever this acetylcholine, function of the acetylcholine receptor will be done, after that it will get destroyed. As a result, there will be like one type of the blind spots. As instantly this mask protein, okay, it will give the intercellular signals, and it will activate certain types of the proteins. There's the kinase means it will phosphorylate. As a result, we'll see the formation of the new proteins that will ultimately produce or from the acetylcholine receptor, that again will get bind with the another acetylcholine that will function here, okay. Now we'll talk about the other etiologies that we see in these cases. Okay. Then the next part, we'll see like some congenital myasthenia gravis. So we can write the congenital myasthenic syndrome. In this congenital myasthenic syndrome, we'll see the patients, okay. Let's suppose there are certain types of the genes that will form these calcium channels, okay, that will form out the sodium channels. Let's suppose the sodium, the calcium, let's suppose is the synaptobremin. There are certain types of the protein that are responsible to form these types of the structures, okay. It is also anti-syntaxin protein. Now for any regions, it's what the acetylcholine receptors. For any regions, if we see there is any type of the mutations, okay, in the genes that will produce this type of the things, if there is any type of the defections or the defective part, if we can find, then this part structurally or functionally will be not enough to do their own functions, okay. So there may be some structural problems due to mutations. If there is some problem for which the calcium will not be able to go within here, open in this part, then if the calcium can't go here, then this part, this part will be not, this syntaxin will be not open. If the syntaxin is not open, then this synaptopepin will not be able to bind with these things. Then it cannot get attached with the membrane. It cannot release the acetylcholine. As a result, there will be impairment with the muscular activities. So, if there is any types of the it's about neurotransmitter synaptic clip. Now, in the problem in these cases, we will see that the persons may have the mutations, the structures of the functions with the presynaptic membrane, the postsynaptic membrane here, postsynaptic cleft or synapses. Okay. Now the problem here we will see in these three parts. One is it may be the problem or mutations in the presynaptic, maybe synaptic or the synaptic cleft. And the third you can write the postsynaptic. Okay. These are the different types of the congenitals. Now we'll talk about the toxins. Hope you understood this part. Now let's talk about some toxins. Okay, so toxin induced neuromuscular junctions functions, impairments of the neuromuscular junctions activity. Now in these cases, especially two types of the toxins we need to know. At least there's a Clostridium. There are many others, but specifically we need to know at least the two. Clostridium botulinum. Okay. Let me write it. Another is tubo with another color. Tuboculare. These two types of the toxins we have to know what's their main function here. Okay. Now the Clostridium botulinum is not. It's the what? It's the gram-positive anaerobic bacteria. Now, what is the function for this thing? Whenever we are eating some types of the home cooked food, and if we see for any regions, a little bit amount of this Clostridium botulinum, it may be like 1 microgram is enough to cause the infections, to cause the harm. Okay. Now, what is the main function here? In the home cooked food, we will see that by chance if this Clostridium botulinum toxin get mixed, then what's the main function will be here? The main function will be see, I already said before that first what? The sodium channels will get activated, the sodium influx, then what? After that, I said you that calcium channels that will activate, and these calcium channels after this activation there will be what? The influx of the calcium within here, within the inner part, and this calcium will unopened part will be opened by the help of this what? Calcium, this unopened part of the syntaxin protein that will be opened, or that will be just like these things. As a result, we will see this part. The acetylcholine will be stored. This part we can be attached with this membrane. Ultimately, it will be able to release the acetylcholine. So in these cases, we can say the calcium dependent releasing of the acetylcholine. Calcium dependent releasing of acetylcholine that will be impaired. Why? Because this Clostridium botulinum toxins will go and they will do the blockage in this part. They will not let the calciums to go in to help in the releasing of the acetylcholine in this part to get bind with this the receptors in the crest part. It's very easy to understand that this Clostridium botulinum they are going and they are getting bind with what? They are getting binded with the calciums channels. As a result, the calcium are not able to go in the motor end plate. As a result, you see they can't help here to remove the but they can't help to remove these parts like the syntaxin parts. This from here. As a result, will not these things will be not able. This acetylcholine or the vesicles, that is stored within the vesicles. Within what the acetylcholine is stored, that will not able to get bind with this part. As a result, there will be no releasing of the acetylcholine, so neuromuscular junctions impairment will be there. So this is on the one part, okay? The Clostridium botulinum. Now, the second part, the tuberculare, okay? In these cases, we will see actually it's like in ancient times we have seen that this type some types of the toxin that was used for hunting purpose. That was used for hunting purpose. Just for example, fishing. Okay. Whenever these things got attached to these fish parts or the fishes, they got paralyzed, they got died, and as a result, they come in the floating. Okay, and these things were collected by these people and they ate it. As a result, these things could go within them. And the main problem here in these parts that they will block this part, the acetylcholine receptors. They will block this part. As a result, no acetylcholine will be able to get bind here. So as a result, ultimately the impairment of the neuromuscular transmissions. Same things here, okay? See the tuberculare. So two types of the toxins, Clostridium botulinum and tuberculare. Now we will see some types of the medicines are there. Those are also responsible for these things. So regarding from the point of view, the medicines, we'll see the just like the aminoglycosides. Within the aminoglycosides, there are various types of aminoglycosides, like we can say like gentamicin, tobramycin. Okay, specifically the gentamicin. The side effects of this gentamicin in some patients, not for all the patients. Okay, otherwise it could be toxins. It could not be a medicine. So in some people or some patients, you can see that these gentamicins will do what they can do the blockage of the calcium channels. They could block this calcium channels. As a result, we will see there will be impairment of in the neuromuscular transmission impairment. There will be dysfunction within this part. Whenever this part will be blocked, calcium will be not able to come here. If the calcium can't come, then what? This syntaxin part will be not open. As a result, this synaptobutrin can get bind with this part. So synaptobutrin will be not able to come get attached with the membrane. So there will be no releasings of the what? This will be no releasing of the acetylcholine. And if the acetylcholine can't come here, then it cannot do its further function. So it can get bind with its receptors. Okay. So just like the medicines, we can see gentamicins. That's the parts. There's in the cluster of the aminoglycosides. There are other some types of the medicines also. Let me write this here and the answer also. Like the quinines, like the beta blockers. Okay, these types of the medicines sometimes we have seen. They are also able, they are having a side effect, little bit side effect in some patients, not for all the neuromuscular transmissions impairment disorders. Now we need to know what is the exact mechanism for the myasthenia gravis. Now the main problem in the myasthenia gravis, we need to know that let me show you again here that Now in this part if we see, then we'll see there is what acetylcholine within the vesicles. There is our synaptoprebin. Now these are the proteins what? Syntaxin, synaptoprebin, acetylcholine. There are sodium channels. There is the calcium channels that will come here. Through this calcium, the calcium will come, it will get bind. This part it will unopened. Then we have seen this will get bind with it. Then from the membrane, there will be releasing the acetylcholine. But myasthenia gravis, I already said you, in 85% of the people, the main problem is the receptors, like the antibodies against these receptors. Okay. Now, so these are the what? Acetylcholine receptors. And against them we'll see the autoantibodies that will be formed. Okay. Basically the IgG antibodies that will form here against it. So the basically we know that IgG antibody is bivalent. It is bivalent. That means what? That can get bind with the two antigens. One antigen will be here, another antigen will be here. That means we can see they are cross-linking with the two antigens, just like this. Okay. Now in our body immune systems, we know that whenever for anything there will be what? The clusters. We will see the clusters, pathological clusters. We will see here the antigen antibody complex, pathological clusters. Whenever these autoantibodies, IgG, that will get bind with these receptors, okay, then a intercellular signal will go within the nucleus. From here, a particular protein will come, and that protein will pull down this whole part. Those proteins are, responsible for the functions of the membranous disorders. After pulling all these things, we will see, this just like this. These receptors, or acetylcholine receptors, they are attaching with this membrane, and of course what? Let me write it here. Okay, with these antibodies. Now, whenever these antibodies will come here, in our cells, the lysosome is there, This lysosome will get attached with these parts. That's why the lysosome is getting attached with this part. And it will secrete different types of the digestive enzymes, especially the protease, or the different types of the digestive enzymes that will degrade all the parts, that will destroy all the parts, like antibodies, Then it will also digest the acetylcholine receptors, or it will destroy all these things. Now the main problem is here, this portion, so this function is done so rapidly. What did I say? There is what? Mass protein also. There is also mass protein receptors that will give the signals. As a result, there will formations again of new what? Acetylcholine receptors. But these functions, this function, the lysosomal activity is so fast that the cell is unable to compensate the productions of the new acetylcholine receptors. The main concept is here that the function of lysosomal activity so fast that our cells can't compensate by producing enough number of acetylcholine receptors. That's the main thing, okay? Actually, these things will be done so rapidly. That our cells can't, compensate these mechanisms. That means our cell is not able to produce enough amount of the acetylcholine receptors, that will get bound with the acetylcholine to do its muscular activity. Because why? Before forming enough amount of what? Acetylcholine receptors, by the essence of the lysosomal activities, all the things will get destroyed. That's the main problem we see here in these cases. Okay. Now we'll see from the different aspects. What type of the problem, others problem we'll see here. Okay. Let me draw here. Now, what type of the other problem we'll see? Let's suppose the muscular part, and here we know that what the acetylcholine receptors is here. Now, whenever this antibodies is IgG antibodies, okay, IgG antibodies will come here, okay. This is the one functions. By this, all the things will get destroyed. Another thing is here that this FC regions of this antibody, that will be activate the complement systems. Within the complement system, we know that this complementary protein that is produced by the livers. So at first we know the C1 complement that will come. After that we'll see the C4b complement that will come. And then it will get bound with the what? C2b, and along the 3 will make what? C3 convertage. They will make C3 convertage. And whenever the C3 convertage will be there, then C3 will get bound. Then this will form this prototype will form C5 convertage. This whole will form the C5 convertage. And whenever the C5 convertage will be just attached here, will be produced, then we'll see the attaching of the C5b. After that, we'll see the further C6, C7, C8, and C9. These all complement proteins will get attached together. Okay. Now, along with that, we have to know that from C5b to C9, this whole protein structures will form a drill type of the functions. They will cause the pore. They will, functions like the drill machines, and they will cause the pores. They will cause the pores just like this, the pores, by this function of these things. Now from C5b to C9, this complex is known as the membranata complex. I'm writing MSC, that stands for membranata complex. And whenever this membranata complex will come here, they will just do the drills. They will form the pores. As a result, we will see the different types of the intercellular materials. Intercellular materials will come from inside, it will come outside, and various types of the extracellular material from here it will go inside. There will be osmotic changes. As a result, we will see the cell will get die. Osmotic changes. There will be change in osmolality. Or the inside material will come outside, or the outside material will go inside. So the cell will get die. This is the function of this complementary fixations, these things. Okay. Now we'll see the just like the steps of the problems in these patients. Now, these are basically the videos related with the pathological of the myasthenia gravis. So, what type of the disorders and how the mechanisms are done.